All right, guys, welcome to your third pre-lab for the Labapalooza labs. This lab is called the color change lab because we're looking at the third sign of a chemical reaction, which is a color change. Normally when a color change happens, it's a sign that there has been a chemical reaction. Some exceptions, though, are when you add a coloring to something, but it stays the same, like when you put food coloring in the food, but it still tastes the same. That's a sign that that color change was not a chemical reaction. Or when you paint something and the color is on top of the surface, but it doesn't change the surface, that's not a chemical reaction because remember, chemical reaction has to create something new. So let's start by talking about safety. Okay, this lab does include some chemicals that can cause some irritation to your skin, so we're gonna wear lab aprons and we are gonna wear goggles again. I know how much you guys love those goggles. So I've got my goggles fitted on. Today's lab has to do with pH, so let me give you a really quick rundown of what pH is. pH is a measurement of the acidity of a solution, and it's measured on a scale that goes from 1 to 14, with 7 right in the middle. 7 indicates something that's not acidic and not basic, it's neutral. The best example of something that's neutral is water. Pure water is neutral, it sits at a pH of 7. Things that are really low pH are acids. Examples of acids include stomach acid, battery acid, lemon juice. The stronger they are, the more they break things down because they're acids. On the other end of the scale are bases. They have a high pH. Bases also break things down the stronger they get. Examples of strong bases include ammonia and lye and things that are used to break things down as a detergent. Oftentimes soaps have a little bit of a high pH. Now if you want to know more about pH, stay tuned. We're going to watch a little 2-minute explanation of how pH works. A question a lot of students have is what exactly does pH stand for? If you're not interested in that at all, just skip ahead 2 minutes and 20 seconds and you'll get to the part of the video where I actually explain what we're going to do today. Okay? All right, guys, so if you want to know more about what exactly pH is, the first thing to understand is that the abbreviation pH stands for power of hydrogen. And by power, what we mean is how much hydrogen is in a solution or an amount of water, and what is the exponent of that concentration, okay? So what that means is as we move from a pH of 2 to a pH of 3, we actually get 10 times more hydrogen. And to make the whole thing a little bit weirder, it's actually negative exponents, but we're going to ignore that for right now. So that's what power of hydrogen means when we say pH. So what does that actually look like? Well, it turns out that acids are really just hydrogen donors, hydrogen ion donors. And bases, they like to give up what are called hydroxyl ions. And so when acids get into water, they release tons of these guys, and when bases get into water, they release tons of those guys. And what happens when these guys meet? Well, that gives us water. So that's why acids neutralize bases, because if you have one of these and one of these, you get water and it goes to being neutral. If you have more of these than you have of these, then these cause it to become more acidic. And if you have more of these, it causes it to become more basic. So that, in a nutshell, is what pH is. pH is just the concentration of those hydrogen atoms illustrated as a 10 to the negative power, okay? And that's why lower numbers actually mean more hydrogen, and higher numbers actually mean less hydrogen. Hope that makes a little bit more sense. All right, so either you skipped over that explanation of pH or you're just more informed now and you know a lot more about what pH is. Either way, to measure the pH of something, you have to use an electronic piece of equipment or a chemistry test, or you have to have an indicator. An indicator is just a chemical that changes color depending on the pH that it's exposed to. So this purple liquid right here is an indicator that comes from cabbage juice. Plants like cabbage and hydrangea, they actually have a color-changing chemical in them that changes colors depending on whether it's in an acid or a base. So for example, over here I have a mild acid, this is some vinegar, and over here I have a mild base. This is stuff that's called soda ash. It's used when you're dyeing clothes to keep the color from changing. If I look at the plain indicator here, this is the indicator when it's neutral. It's just mixed with plain water. And you can see the color that indicates a neutral solution is this purple color. So when you see the cabbage juice indicator purple, you know that it's neutral. If I add it to the acid, you see that it turns a pink color. Reddish pink indicates an acid. And if I add it to the base, it turns a bluish turquoise color. That suggests a base. Now the way that I remember that is I think blue equals base, and whatever the other color is equals acid. So this is a low pH, a high pH, and a neutral pH. And remember that one of the critical parts about pH is that when you take an acid and a base and you add them together, you put them in the same container, they neutralize each other, they cancel each other out. It's like a game of tug of war. And if you can get the rope to stay right in the middle, that's neutral. So that brings us to our lab. In the lab, you're going to be given a cup of indicator, which is that purple color because it's neutral, and you're going to be given a clear liquid that is an acid, and a clear liquid that is a base, and a clear liquid that is an unknown. Now, each team will get a different unknown, so you won't know what the unknown is. That's why we call it the unknown. Your job is to take the pipette and add indicator to the acid and verify that it's an acid by getting that color that you're expecting. Then add some indicator to the base. Make sure you get the right color in the base. It's really important when you do that you suck up liquid with the pipette, but don't put the tip of the pipette into the liquid, because if the tip of this gets into the acid and then it gets into the base, you're actually putting acid into the base, and that's going to mess up your experiment. Then you're going to add some indicator to the unknown. And that should tell you whether the unknown is an acid or a base. So question number one that you have to answer is the unknown an acid or a base? And you'll know that as soon as you add some indicator to it. Then comes the fun part. The next thing we're going to do is called a titration. If the unknown is an acid, we're going to add the opposite of an acid, which is a base. And if the unknown is a base, we're going to add the opposite of the base, which is an acid, until we get it back to neutral, okay? So you're going to want to count how many drops of the opposite it took to get it back to neutral. And that's how we measure how strong an acid or a base is how many drops of the opposite it takes to get it back to neutral. So question 2 that you have to answer in your notebook is, how many drops of the opposite did it take to neutralize your unknown, okay? And then the last question, question 3, in your notebook, you have to explain to me, what is the pH scale? What do high pHs, low pHs, and middle pHs mean? Again, what is the pH scale? What do low pHs, high pHs, and middle pHs mean? That's it. Enjoy.